Okay, so I want to go over this paper called Galectin-3 Binding Protein, which is a multitask glycoprotein with innate immunity functions in viral and bacterial infections. So the reason why I want to go over this galectin-3 is because we do know that the galectin-3 binding protein is upregulated in those with COVID-19, and the more galectin-3 binding protein that is being produced will be a biomarker for more severe COVID-19 disease. So this is at least in the plasma of people who have COVID-19. So I want to go over this article to help us gain a better understanding as far as what its role in the body is. So with that, the abstract, galectin-3 binding protein is a ubiquitous, and ubiquitous meaning it's found all over, and it is multifunctional secreted glycoprotein, which would be a protein that has glycans on it, or carbohydrates that have been added onto the protein. And this was originally identified and mainly studied in the context of neoplastic transformation and cancer progression. So neoplastic transformation, this is the when the cells are turning into cancerous cells. So yeah, neoplastic transformation involves 4 sets of genes: the oncogenesis, the tumor suppressor genes, the mutator genes, and the apoptotic genes. Don't worry too much about that. Just know that that is the process of which a cell will undergo to become a cancer cell. However, galectin-3 binding protein expression is induced in viral infection and by a multitude of molecules that either mimic or are characteristic for an ongoing inflammation and microbial infection. And a multitude of molecules, these are the multitude of molecules. So the interferon alpha, the interferon beta, the interferon gamma, the tumor necrosis factor alpha, the poly-IC. I'm not familiar with what that is. Let's look that up real quick. If our Google polyinosinic polycytidelic acid. So what is this? So this is an immunostimulant, and it is used in the form of its sodium salt and to simulate viral infections. So it's known to interact with these toll-like receptors, which are a type of receptor on your cells, which are expressed at the endosomal membrane of B cells. So the B cells have these toll-like receptors in their endosomes, which are small little bubbles within the B cells, or the macrophages, or the dendritic cells, which are those are other types of immune cells. So poly-IC is structurally similar to double-stranded RNA, which is present in some viruses and is a natural stimulant of the toll-like receptor 3. And thus, poly-IC can be considered a synthetic analog of double-stranded RNA and is a common tool for scientific research on the immune system. So the reason why polyinosinic polycytidelic acid is going to stimulate your immune system is because within your body and with the bodies of all eukaryotes, really, RNA isn't normally double-stranded. So if you see double-stranded RNA, that usually means that it's coming from some form of pathogen, such as a virus. So remember, your genetic material can be DNA or RNA, and both DNA and RNA can exist as single-stranded or double-stranded. And so because of this, since all the DNA in your body is double-stranded pretty much, and all of the RNA in your body is single-stranded, when you see a double-stranded RNA, that would be a way to indicate to your cell that this is not good, this is from a foreigner, not from you. So this poly-IC mimics what that double-stranded RNA does. So yeah, sorry for going on that tangent. So there's also double-stranded RNA and then also double-stranded DNA as well, which would require different mechanisms of attack. So furthermore, galectin-3 binding proteins belongs to the scavenger receptor cysteine-rich domain-containing protein family. So a lot of freaking words right there. So What is the scavenger receptor cysteine-rich, or SRCR? Well, it is a conserved domain. Let's get a Wikipedia article if they have it. So this is the domain where it has a lot of cysteines, which cysteines are amino acids that have sulfur groups on them. And so the SRCRs is short for this scavenger receptor cysteine-rich domain, and they are found solely in eukaryotes. And these domains are present on the cell membrane and have a role in binding to specific ligands or things that are floating around in solution that will reach a receptor. And these are often found to be involved with the immune system. So just so you know, this is what this type of domain is, and there's a lot of variety in the amino acid sequences that can create this domain, but they are all related to each other. So this Galectin-3 binding protein, which I'll get a Wikipedia page for that as well. It looks like this, which, you know, if you squint your eyes, it might look similar to you. However, just know that this Galectin-3 binding protein is a member of this SRCR family, okay? And yeah, if you go back and forth, you can definitely see that there is a lot of similarity in the structures of them because they are related to each other. So furthermore, and by virtue of its N-terminal SRCR domain, and the SRCR domain is found in soluble or membrane-associated innate immunity-related proteins. So this SRCR domain is found both in the solution and anchored to the membranes of cells, and it is implicated in self versus non-self discrimination. So these scavenging receptors are able to somehow figure out how to separate between what are proteins that your own body produces and what are proteins that the pathogens are producing. So this review summarized the current knowledge of structural features of the Galectin-3 binding protein and its proposed intracellular and extracellular innate immunity functions with special emphasis on viral and bacterial infections. So I like how it's talking about what is going on intracellularly or within the cell and what is going on extracellularly. So if you watch my previous video, we talked about not Galectin-3 binding protein, but Galectin-3. And Galectin-3 will actually have completely different functions based on whether it's inside of the cell or outside of the cell. And at least when it comes to the T cells, which are a subset of the immune cells, some T cells are protected by Galectin-3 when the Galectin-3 is inside of their cytosol or inside of them. However, when Galectin-3 is on the outside and interacts with the receptors on the outside, that actually convinces those T cells to kill themselves. So basically, you could have the opposite function depending on where these proteins are located. So just keep that in mind, that when we are talking about proteins, the location is important. So sometimes we might say, oh, this protein has this function. Oh, well, shoot, it actually has the exact opposite function in other scenarios. So it's really difficult to try to drive points across when you have to deal with completely opposite effects of the same protein. So let us go down. So Galectin-3 binding protein, and this is the UniProt ID if you want to go to uniprot.org and look it up for yourself. This is also known as 90K or MAC2 binding protein, and these are remnants of when Galectin-3 binding protein was first discovered. It was 90 kilodaltons, and then it also binds with this MAC2. So that was, we have a million different names for every single protein, and a lot of that comes from the experimentation that was done to discover them in the first place. And then they get renamed over and over again. So anyways, this was originally described by several independent research groups while aiming to identify proteins secreted in vitro by cancer cell lines, such as CG5, so breast cancer. So frequently we do experiments with cancer cells because the cancer cells can duplicate indefinitely. And so we can use these cancer cell lines for research on cells that would be difficult to do on normal living tissue. For one, there's very little, or it's more difficult to be reproducible when you're using living tissue because who knows what was going on in the original human. And then also, it's difficult to do research on human subjects because it's very difficult to find human subjects. So, anyway, CG5 would release more of this galectin-3 binding protein, and there's also this Calu-1, which is a lung cancer, and they identified that they identified this being secreted by these cancer cell lines utilizing a monoclonal antibodies, so just using antibodies to discover whether or not a protein was present. Alternative lines of investigation focused on cellular or secreted proteins that interact with beta-galactoside binding lectin, or Galectin-3, Gal3, right? So, such as from melanoma cells, such as from the melanoma cell line A375, various colorectal cancer cell lines, and breast cancer cell lines, or cell line SKBR3. So, their galectin-3 is implicated in a lot of other types of cancers when that gets dysregulated. So there was research being done to figure out what proteins bind to galectin-3 to help figure out the whole signaling cascade of these different cancers. And so this is why we have multiple names for the same protein, because the same protein was appearing in several independent research groups looking up different things, and so they all come up with this name for this factor or this name for this protein, and then later they discover, oh, we were all actually working on the same protein, so we need to consolidate all these names now. So primary motivation for many of the early Gal3 binding protein studies was not only to identify proteins involved in neoplastic transformation and cancer progression, but also to identify diagnostic and prognostic markers as well as druggable targets. So when we do these studies, it's not just to know more, although that is virtuous in of itself. We also do it to identify diagnostic tools. So if we take a blood sample or if we take a sample from whatever cancer you have, and then figure out what proteins are being expressed in it, this could clue us in as to explicitly what type of cancer it is, or if you have cancer in your body. Another thing would be if these biomarkers are playing a role in the cancer progression, we might be able to develop drugs that target that particular protein, and by targeting that protein, we might be able to up or down regulate its activity, which could result in preventing the progression of the cancer or killing the cancer if that's possible. So high serum concentration, so serum of the blood serum. The high serum concentrations of Gal3 binding protein were detected early on in patients infected with HIV and hepatitis virus, and more recently, patients infected with dengue virus, Epstein-Barr virus, and Pumala hantavirus. One recent report describes elevated serum Gal3 binding protein concentrations in patients suffering from blood culture positive bacterial infections. So you can see that this galectin-3 binding protein is being upregulated into the serum somehow, and this is being done frequently in conjunction with either a viral or bacterial infection. Excuse me. Thus, it appears that galectin-3 binding protein production is upregulated in response to a microbial infection. This review summarizes the current knowledge of structural features of galectin-3 binding protein and then focuses on its proposed role in innate immunity against viral and bacterial infections. So when we're talking about innate immunity, we're talking about the immune response that is not, sorry about that, we're talking about the immune response that is not specific to a specific disease. We're talking about, so for instance, the adaptive immune response would be an example of your antibody creation, where you're creating antibodies against a particular disease that has infected your body. Well, the innate immune response would be the different types of things that happen within the cell that protect the cell from viral pathogens or other pathogens, or things dealing with, general inflammation or general cell signaling that says, hey, you need to worry about this area because there is a pathogen nearby. So those would be a form of the innate immune response, whereas the adaptive immune response would be when you have, say, developed antibodies against a particular disease that is able to get past your innate immune response. That is what you're going to need antibodies for, because your innate immune response in of itself is not strong enough to prevent that disease from taking over you. Anyways, the structural features of Galectin-3 binding protein. So several independent research groups contributed to the characterization of the Galectin-3 binding protein encoding gene, which is LGAL-S3BP, from humans, mouse, and rat. The human gene locates in chromosome 17 quarter 25. I don't know exactly. This is just a part of chromosome 17, and it has 6 exons, which are transcribed into mRNA to yield a protein of 585 amino acids, and you can see this with this Uniprot ID. So mouse and rat LGAL S3 binding protein encode theoretically for a protein of 577 and 574 amino acids, respectively, and are nearly 90% identical to each other. All 3 proteins share the same domain structure as shown in Figure 1 for the human and mouse Gal3 binding protein. So here they're just saying that the DNA of humans, mice, and rats will encode for 3 different proteins, and those proteins are of different lengths, but they are pretty darn similar to each other. So several reports show that both secreted and cell-associated human Gal3 binding protein lacks 18 amino acids from its N-terminus, as compared with the nucleotide sequence-based translation of LGAL-S3BP. So we can figure out, based on the DNA of a gene, we can determine what how do I phrase this? We can pretty much do a really good job at determining what the amino acid sequence should be when it is first transcribed into mRNA and then translated into the final protein. So from the DNA code, we can sort of figure out what the initial translated protein is going to end up as. But that will not tell us, say, if there are post-translational modifications or different types of modifications that happen to the protein after it's synthesized. It's more difficult to be able to predict what those are, just because that's not an easy computer program. That will be context dependent. It will depend on what cell you're talking about. That will depend on what chemicals, what cellular signaling. It'd be the whole nature versus nurture argument. The nature would say this is what the protein is supposed to be, but the whole nurturing aspect would be all these different proteins that are floating around in solution of the cell that are going to be modifying that particular protein over and over again until it's in its final mature form. So this is consistent with a protein maturation process involving a proteolytic processing of an N-terminal signal sequence. So the N-terminal would be at the beginning of the amino acid sequence. So there's very often going to be there's a signal on the N-terminal side of proteins, and then a lot of times that signal is going to be cleaved off during processing. So that seems to be what has happened to Galectin-3 binding protein. So, and this is guiding the protein into a secretory pathway. So this is a very, very common for secreted proteins. So secreted proteins being ones that end up going to the cellular membrane or being secreted out into the environment. So following the signal sequence, Galectin-3 binding protein contains a scavenger receptor cysteine-rich domain, which is the domain we were talking about earlier, and making it a member of the SRCR protein superfamily. The SRCR domain is an ancient and highly conserved domain of about 110 residues, which is found in soluble or membrane-associated innate immunity-related proteins, such as complement factor 1, deleted in malignant brain tumor 1 protein. So this is this would be a protein that, if it is removed, will cause a lot of brain tumors. So oftentimes we name a lot of proteins after the diseases that they cause if the protein isn't working correctly. So, and this is a recurring theme in a lot of molecular biology. So, and then also there's a cluster of differentiation 5, cluster of differentiation 6, and CD163, which, what is CD163 again? Is a high affinity scavenger receptor for the hemoglobin heptoglobin complex. Okay, so yeah, I've seen this before, but I didn't know exactly what it does. I'll probably have to read more about that later. Anyways, the SRCR domains associate with protein-protein and protein-ligand interactions, including direct interactions with microbial surface structures. For example, DMBT1 GP340 binds to, and remember that's the deleted in malignant brain tumor 1 protein. This binds to the leucine-rich repeat-containing surface proteins of both gram-positive and negative bacteria. So in gram-positive and gram-negative bacteria, don't worry too much about that. Just know that these are types of bacteria. They will often have these leucine-rich, which leucine is one of the amino acids. It's very hydrophobic. There will be leucine-rich repeat-containing surface proteins on both the gram-positive and negative bacteria. And because this is such a common thing on some of the proteins of these bacteria, it would be beneficial to have a receptor that can recognize this particular design, if you will. So, and by recognizing that, it is independent of what type of, it would be part of the innate immune response, because this doesn't require antibodies, right? This doesn't require a finely tuned adaptive process to create these antibodies against these particular bacteria. They already have a receptor that has the ability to recognize that this is not necessarily good to have in your body. Anyways, CD163 mediated recognition of gram-positive and negative bacteria leads to the pro-inflammatory response. So this would be a way CD163 would be able to recognize these bacteria and then cause an inflammatory response, which theoretically, or which would end up bringing more immune cells to the site of infection, which then you could theoretically develop a more robust adaptive immune response if the need were to arise. So, and also those white blood cells will also be attacking cells that have the, what would the word be? They're just going to just search and destroy, right? So they're going to do a lot of different things to help fight the infection for you. Excuse me. So CD5 has been reported to recognize 1,3-beta-D glucan-containing fungal cell wall components and to regulate the antifungal immune response. So you will have these CD5 receptors, and these CD5 receptors will be very good at detecting this 1,3-beta-D glucan carbohydrate. So this carbohydrate is very common on the fungal cell walls, and so this CD5 will recognize those particular glycans, and by discovering those glycans, it can then warn everyone, hey, there is a fungus here, we need to kill it. So the crystal structure of the Gal3 binding protein SRCR domain has been determined to 2 angstroms, and angstroms is just 10 to the negative 10th meters resolution, and it shows a compact fold of 6-stranded beta sheets and alpha helix and 3 intramolecular disulfide bonds. So remember when we were talking about this protein domain, or this protein LGAL S3BP, well, this is only part of the whole Galectin-3 binding molecule. So this is just the SRCR domain. So here we have the SRCR domain, the scavenger receptor cysteine-rich protein domain. Well, that is only the part of the Galectin-3 binding protein that has that domain in it. Only that domain is shown right here. there is more to this galectin-3 than what is pictured right here, is what I'm trying to get at. So for long, this was the only SRCR domain structure available. But currently, several SRCR domain structures are known. For example, the Marco, CD5, CD6, and they all show a fold similar to the galectin-3 binding protein, with most of the secondary elements conserved. So in addition to the SRCR domain, galectin-3 binding protein harbors a BTB pause, or the broad complex, tram-track, and bric-a-brac, poxvirus, and zinc finger domain. So, freaking biology, right? We love our acronyms. Anyways, it has that, and it also has a BAC, which is a BTB and C-terminal Kelch domain and approximately 200 amino acid C-terminus with no significant similarity to other proteins. So what I'm going to say is look at this acronym. Well, this acronym has another acronym inside of it, right? So this BAC has BTB and C-terminal Kelch domains. Well, that BTB also is this broad, complex, tram-track, and bric-a-brac. Yeah, we'll just leave it at that. We love our acronyms in biology. So let's talk about the BTB pause domain. So these domains are found in proteins involved in a wide variety of biologic processes involving transcription, regulation, cytoskeletal rearrangement, and protein ubiquitination. So cytoskeletons, this would be the skeletons of the cell. This would be just different protein. It would be like the protein web that helps move things around and gives the cell structure, right? So actually, I believe it's on the. Let's look up what this is so that we can have a better understanding of it. So the cytoskeleton is a complex dynamic network of interlinking protein filaments present in the cytoplasm of all cells, which is including bacteria and archaea. And it extends from the cell nucleus to the cell membrane, and it is composed of similar proteins in various organisms. In eukaryotes, it is composed of three main components: the microfilaments, intermediate filaments, and microtubules. So this would be an example of this colorful green would be an example of all of the microtubules that is a part of the cytoskeleton. And then there's also the actin filaments, which are shown in red, which are also part of the cytoskeleton. So just so you know, those are just the proteins that help give the cell its rigidity and allow things to move around within it. Anyways, the cytoskeletal rearrangement is frequently associated with proteins that have this BTB-POZ domain. And then also there's the protein ubiquitination, which is ubiquitin is a small protein that is frequently used as a tag to put onto other proteins. And so this tag of ubiquitin can then warn the rest of the cell what to do with this particular protein. Sometimes it's to signal that the protein needs to be degraded, but that is not the only thing. There's a lot of different things that ubiquitin can do. Anyways, this BTB pause domain often self-associates as dimers, tetramers, or sometimes even larger oligomers. Indeed, purification of Gal3 binding protein from human secretions and cell culture medium showed the protein to form high molecular mass oligomers with ring-shaped structures of 30 to 40 nanometers in diameter. So it seems as though this galectin-3 binding protein is able to form rings. And by forming these rings, they might theoretically have a completely different function from the monomeric form. Anyway, scanning electron microscopy mass analysis indicates that oligomers are formed by even numbers of monomers, and the dodecamers are the most prominent. So having 12 of these come together to form a ring is the most prominent type of ring that they see. Ultracentrifugation, electron microscopy, and recombinant truncated proteins serve to resolve the role of different domains in the ring formation. So yeah, just different experiments were done to try to figure out the ring formation. The SRCR domain does not self-associate. So this SRCR domain in of itself is not able to form these rings. But a truncated form with both the SRCR and the BTB pos domain, these form exclusively dimers. So if you only have these two parts of the protein, it will only form dimers. It won't form those rings, right? A truncated form with the BAC domain and the C terminus form heterogeneous aggregates. So they'll basically form very amorphous blobs, right? If you just have the back domain and the C terminus. And the SRCR lacking Gal3 binding proteins both form both homopolymers and rings. So if you have the rest of the Galectin-3 binding protein without SRCR, apparently you can form both homopolymers and rings. So those slightly unstructured, so they don't have the same structure if they're missing the SRCR. Thus, the BTB POZ domain of the Galectin-3 binding protein appears to mediate dimer formation, but all domains are needed for the correctly structured ring-shaped oligomers. So everything is playing a role in this particular protein to form these dodecamers. Now, granted, there are a lot of proteins where that will form polymers, and you can remove entire domains from those proteins and they will still form their polymers, but this is not one of those proteins. This is one of the proteins that will require all of its domains to properly form these ring-shaped structures. Anyways, several studies showed that Galectin-3 binding protein purified from cell lysates, so from the inside of the cell, in cell culture media, as well as from human milk and serum containing 2 to 3 distinct forms with approximate sizes of 90, 75, and 25 kilodaltons. Okay, for example, N-terminal sequencing of the different fragments revealed that the sequence between the back domain and the C terminus is prone to proteolysis. So what they're saying here is that with this particular protein, in many different fractions, so in the cells or in the cell culture media or in even human milk, you can find this Galectin-3 binding protein to be in different sizes. So this is frequently because of proteolytic cleavage, where the protein is being broken down by a protease somehow in its maturation process. So with that, just know that there is this pretty common cleavage site that is between the BAC domain and this C terminus. And they call it the C terminus because there is no known, they haven't figured out what type of domain it is yet. So it might not even be that well structured. So indeed, a brief treatment with plasma, lyse or trypsin, so these are different proteases. This results in the formation of approximately 25 kilodalton fragment cleaved after amino acids 442 or 455. So let me get this uniprot. I recently did an alignment with, and let me get the similarity turned on. I recently did an alignment with the SARS and the original SARS and SARS-CoV-2 spike proteins, and I did that alignment with this binding protein. So I think I have more to talk about that later, but what I want to focus on is that here are the results. So in this location, and let's say this is between amino acid 442 or 455. Well, here is 442 on this human, the human Galectin-3 binding protein. So this is where it's at, and it actually seems to correlate pretty well with an area that has a lot of homology with the SARS coronavirus. And I misspoke. This wasn't the spike protein, or if I did say the spike protein, that was wrong. So this is not the spike proteins of SARS-CoV and SARS-CoV-2. This is the R1AB protein, which is one of the main proteins that gets proteolytically cleaved into the different subunits to produce the RNA-dependent RNA polymerase, and I think a few other proteins as well. So let's see if there are known cleavage sites that I need to be aware of. So repeat. See, let's look for. Yeah, I don't think there is a good. Maybe domain. Nah, that won't be a good one. So yeah, don't worry too much about this. I'll probably do another video if I can think of more later. Just know that there is a cleavage site right there, and then there is also another cleavage site that is in 455. So there'd be another cleavage site around here, right? So actually, so 444. 445, 446, 47, 48, 49, 50, 51, 52, 53, 54, 55. So there's another cleavage site right here as well. So what this would indicate to me, and this spot probably cleaves easily because of this arginine that is located here, but I want to bring attention to the fact that these YRY sequences, these are frequently, what would the word be? These are frequently carbohydrate mimicking sequences. So these are sequences of protein that sometimes look like carbohydrates to different enzymes, and they can actually interact with different lectins, which lectins are carbohydrate binding proteins, but this particular sequence seems to be able to confuse a few lectins. So just keep that in mind, that these cleavage sites seem to be pretty darn close to where these carbohydrate mimicking peptides are at. And what I find very interesting is that we have the repeats of these these tyrosine rich domains. There seems to be correlation between what is happening in Galectin-3 binding protein and what is also seen in these R1ABs from these coronaviruses. So part of me wants to dig into whether or not these coronaviruses are actually mimicking some carbohydrates in order to modulate the immune response in some way. So just keep that in mind, that this is probably a reality, because this is a very this would be indicative of very effective carbohydrate mimicking. Regardless, so let's continue. And this isn't, oh, I should also say, this isn't to say that this R1AB of the coronaviruses, it's not to say that they, somehow stole this motif or they stole this domain from Galectin-3 binding protein. It might have been simply that this is a very useful motif to form, and so they both the Galectin-3 binding protein and the SARS coronaviruses created this motif or evolved this motif independently in order to exert its effects somewhere. So just keep that in mind. This doesn't mean that it stole it. It just, or that they are related. It just means that they have similar amino acid structures because whatever function they're doing is probably similar. So sorry, I've been rambling. So let us continue down to, so here, this is where the cleavage happens. So, and endogenous proteases of breast cancer cell lines, SKBR3, they cleave the Galectin-3 binding protein after amino acid 437. So that's also pretty close to that. However, Galectin-3 binding protein can be purified from cell culture medium as a single homogeneous form of approximately 90 kilodaltons in size if protease inhibitors are used throughout the Galectin-3 binding protein purification protocol. So if you are trying to purify the Galectin-3 binding protein from cell lysates, or the insides of the cell, if you use proteases the entire time, the galectin-3 binding protein remains intact. So it has been hypothesized that proteolytic processing of the C-terminal regions of galectin-3 binding protein may have regulatory function. And I'd believe that. I really would, especially since it is close to that, to those carbohydrate mimicking motifs. So the calculated molecular weight of human galectin-3 binding protein monomer is 65.3 kilodaltons, but the secreted form of a protein usually appears as a 90 to 100 kilodalton protein in SDS page, and this is sodium dodecyl sulfate polyacrylamide gel electrophoresis. That is, just know that SDS, this is a detergent that completely denatures the protein and then also gives it a uniform charge. So, the proteins will have different charges, which make it very difficult to control for, unless you just completely destroy the structure of the protein and you give it all a uniform charge. Then you can run this charged particle through a gel, and you are actually using a voltage to move the charged proteins through that gel. So the voltage is moving those proteins. However, the size of the protein will determine how quickly it will actually move through that gel. So if you use more voltage, it will move the proteins faster. However, there might be a bigger differential of, like, the voltage might not be as uniformly distributed through among the gel if you increase the voltage, and so you can actually have the protein not move uniformly. So just know that's what SDS page gel is doing. Oh, and I should say the gel is made out of polyacrylamide, so that's where the PA comes for in page. So what was I getting at? So if you're supposed to, the monomer is supposed to be 65.3 kilodaltons. However, in SDS page, we keep on seeing 90 to 100 kilodaltons. And so treatment of human galectin-3 binding protein with N-glycosidases reduces the molecular mass of the secreted protein back or near to the calculated value. So these N-glycosidases, these are going to be removing sugars from proteins, explicitly from nitrogens, which nitrogens are a common atom at which glycosylation will happen on a protein. But this N-glycosidases will remove those sugars, which then will revert this 90 to 100 kilodalton protein back to this 65 or close to the 65.3 kilodalton protein, which tells us that most of the size difference is due to N-glycosylation. So just keep that in mind, that the protein normally is heavily glycosylated. So treatment of human galectin-3 binding protein with N-glycoside, oh wait, I read that. So this implies extensive glycosylation. Indeed, the sequence of human galectin-3 binding protein contains seven potential N-glycosylation sites. Partial N-glycosylation or partial N-glycosides treatment experiments suggest that all 7 N-glycosylation sites are occupied. So basically we can do a good job at predicting what nitrogens are likely going to become glycosylated, and if we do partial glycosylation of galectin-3 binding protein, it appears as though that those 7 distinct sites, each and every one of them, have glycosylation that is on them. So yeah, that I would presume that the experiment would be done in such a way that if you did it, you could see a distribution of different weights, and that distribution of different weights would indicate how many N-glycosylation sites there are. So if you could imagine, if there was only one of those N-glycosylation sites that was glycosylated, and you did partial treatment, you would only see two bands. One where that nitrogen was glycosylated, and one where that N-glycosylation did not happen, right? Or where they removed it. If there were, say, 2 of the 7, then you would actually have potentially up to 4 different peaks, if you will, because then you would have one where they're both glycosylated, one where only one is removed, one where only one is removed, and one where both of them are removed. So I haven't seen the experiment for myself, but that would be my presumption that this is how they would figure out that all 7 of the N-glycosylation sites were actually being glycosylated, because there's probably 2 to the 7th power of different peaks that they saw. That would be my guess. Anyways, sialidase treatment. So sialidase, this is, well, sialic acid is one of the glycosylations that could theoretically take place. So they cut the glycosidic linkage of neuraminic acid. So this would be N-acetyl neuraminic acid, and if you had this OH over here, could theoretically be used to form a glycosidic linkage with some other organism, or I should say, or with some other protein. But what I should say is this carbon that is connected to this oxygen, this carbon theoretically could form another bond with a different oxygen or nitrogen. So if it forms a bond with nitrogen, that would be an N-glycoside. If it forms a bond with a new oxygen, that would be an O-glycoside. So yeah, I think, that's all that I need to really say for this. And theoretically, it doesn't need to be a protein. I just said protein, but I'm not as familiar with all this stuff, and I should look it up more, but it's not important to me right now. So sialidase treatment and lectin binding studies. So lectin, these are the proteins that can bind to sugars. So the sialidase treatment and the lectin binding studies also show the presence of terminal sialic acids and fucose in some forms of galectin-3 binding protein. So what this is saying is, if you treat your galectin-3 binding protein with sialidase, this is telling you that you explicitly have sialic acid that is on the protein. So remember up here when we were talking about N-glycosidases, this wasn't as specific, right? This is just saying, oh, you have sugars. But here with the sialidase, we're saying if you use sialidase and you're removing explicitly sialic acid from your protein, your lectin is not able to bind it in the same way. So that's what these lectin studies are doing. And so this is telling us that there is the presence of sialic acids and fucose, which is in some forms of galectin-3 binding protein. And fucose is just another, sugar that can frequently be found on proteins. So O-glycosylation does not exist or has only a minor role. So remember how I said you could have both N-glycosylation or O-glycosylation. That is the difference between having a nitrogen be glycosylated. Sorry, the UPS struck. So that would be the difference between having nitrogen being glycosylated or an oxygen being glycosylated. So this does not exist or only has a minor role in galectin-3 binding protein. The oxygen form doesn't. So the mouse galectin-3 binding protein, also known as CyCap, is also a secreted protein which goes through similar maturation processes as human galectin-3 binding protein, involving post-translational N-linked glycosylation from an initial 64.4 kilodalton cell-associated form to an approximately 90 kilodalton secreted form. So frequently, this protein will be processed in order to become secreted, right? So very frequently, glycosylation is necessary for a protein to become secreted and to be, for one, even be soluble enough to go out into the serum of your body, so that it can float around and then find its potential targets. And there's, I would also assume that the cell-associated form, which I believe has a GPI anchor on it, that I believe that would also require some glycosylation. So the mouse galectin-3 binding protein has 6 potential, N-glycosylation sites. If all the 7 N-glycosylation sites of the human galectin-3 binding protein are mutated, or cells are treated with tunicamycin, which is a chemical inhibitor of N-glycosylation, very low amounts of the protein is secreted. So if you do not have the ability to glycosylate your galectin-3 binding protein, that is going to end up staying within the cells and it is not going to be secreted into the environment. So this is not surprising given that the secretion of galectin-3 binding protein involves an interaction with the ERGIC53. And this is an endoplasm reticulum Golgi transporter molecule that recognizes high mannose type and glycans. So just know that this ERGIC53 is in the endoplasmic reticulum and Golgi apparatus, which is where a lot of proteins end up getting synthesized into. So they have, at least the rough ER has these ribosomes that are on it, and then proteins are frequently translated and injected directly into the endoplasmic reticulum. And then there are a lot of enzymes and stuff within the endoplasmic reticulum that end up doing post-translational modification of the proteins. Frequently, well, I believe all secreted proteins really go into the endoplasmic reticulum and get processed so that they can be secreted. But one of the enzymes that are necessary for this proper secretion is this ERGIC53, and the way that it works is that it recognizes these high mannose. Where is this at? It recognizes these high mannose type N glycans. So mannose is just another sugar. So there's hundreds, if not thousands, of sugars depending on your definitions of them. So mannose is just another one of those sugars. And if you have a lot of these mannoses on type N or attached to nitrogens on your proteins, that will be recognized by ERGIC53, and then it will transport those proteins somehow. So thus, N-glycosylation of galectin-3 binding protein appears to be essential for its secretion, in addition to its role in the galectin-3 binding protein interactome. So the interactome is just the fancy word for saying what other proteins this galectin-3 binding protein will interact with. So 53. So expression patterns of galectin-3 binding protein. So early northern blot studies showed an abundance of this binding protein mRNA. So the mRNA of the galectin-3 binding protein, which the mRNA, as I'll probably repeat over and over again, since it's the central dogma of biology, the DNA gets transcribed into RNA or messenger RNA, and then that messenger RNA gets translated into the final protein. So here we're talking about the mRNA that is going to be translated into this galectin-3 binding protein. So this signal, early northern blot analyses, and northern blot would be the visualization of the different types of mRNA that is found in a sample. So early northern blots studies showed abundance of the mRNA of the galectin-3 binding protein in many normal human tissues, and especially prominent expression was seen in the lung and gastrointestinal tissue. So this is a protein that is found throughout the entire body, or at least in a lot of different tissues. However, there is a lot more being expressed in the lung and gastrointestinal tissues. So what I'm going to do is I will, let's see if there is so this would be an example of the quantification of the mRNA that is in the different cell types. And as you can see, there seems to be a lot of different cells within the human body that can produce this galectin-3 binding protein. However, it is very high in, say, the lungs. And so it looks like the heart produces a lot of it, the placenta, the lungs, and the prostate, and the thyroid. Those produce a lot of it. And then obviously there's quite a few other tissues that produce a lot too, but these are the most. Anyways, that is an example of the mRNA quantification found in a lot of different proteins. So This is in accordance with the tissue expression patterns found in the Human Protein Atlas, which is a freely available interactive data resource of samples representing all major human tissues analyzed by immunohistochemistry, don't worry too much about that, and the RNA sequencing. So, in that data set, the Galectin-3 binding protein and the LGAL-S3 binding protein mRNA, these are found at variable levels in all tissues. So high expression is seen, for example, in gastrointestinal tract and lung, whereas relatively low or nearly absent Galectin-3 binding protein is seen in the brain and pancreas. So we normally have very low abundance of this Galectin-3 binding protein in the brain and the pancreas. That is interesting. I might have to come back to that later. So it seems... So Galectin-3, so not the binding protein, the original Galectin-3, I believe plays very important roles in neurodegenerative diseases. Yeah, so it's very important in this Galectin-3 is very important in myelination. So okay. I might have to do more reading on all that later. So the Galectin-3 binding protein is found in the serum and in other bodily fluids, so of apparently healthy individuals. However, the Galectin-3 binding protein serum concentrations and expression levels of Galectin-3 binding protein in tumor tissues, these are significantly elevated in several neoplastic pathologies and predict poor clinical outcome, for instance, in breast cancer and lung cancer. However, also contradicting findings exist, for example, in breast cancer, which could relate to the strong negative correlation detected between expression of Galectin-3 binding protein and ERB2 receptor tyrosine kinase. So a known marker for aggressive progression of breast tumors and poor prognosis. So let me read this again. However, also contradicting findings exist, for example, in breast cancer, which could relate to the strong negative correlation between expression of Galectin-3 binding protein and this ERB2 receptor tyrosine kinase. This ERB2, if it's very upregulated, if I'm reading this correctly, would be very bad or at least would result in breast cancer. So, and this is a known marker for aggressive progression of breast tumors and poor prognosis. So it could be that this ERB2 is negatively affecting Galectin-3 binding protein. So in fact, a positive prognostic value for Galectin-3 binding protein has been reported in certain neoplastic pathologies, such as in colorectal cancer and Ewing's sarcoma. Galectin-3 binding protein has also been proposed a role in systemic autoimmunity. So Galectin-3 binding protein concentration is highly elevated in circulating microparticles of systemic lupus erythematosus patients, and the concentration strongly correlates with disease severity. Now, this is where I need to also mention is that this means that they're correlated. So, for instance, if Galectin-3 binding protein is providing a very protective effect or a protective role on the body, it could very well be that people with cancer have high levels of this because it's trying to protect itself. It doesn't necessarily mean that the Galectin-3 binding protein is causing, or at least being dysregulated in it. It could be very well that it's upregulating in order to protect you. Granted, it very well, since Galectin-3 binding protein is also associated with the pro-inflammatory response, that it might be that it is being upregulated in order to further the cancer. So it's really hard to keep all this straight sometimes, but just know that that might be happening. Also, circulating Galectin-3 binding protein concentrations are increased in juvenile idiopathic arthritis patients compared with healthy individuals. So, as explained in detail below, the Galectin-3 binding protein expression and secretion are also significantly upregulated in various microbial infections, indicating a role in innate immunity against viral and bacterial infections. So, Galectin-3 binding protein and innate immunity. And I'll probably be able to finish this within one video. So, molecules that induce galectin-3 binding protein expression and secretion. So, first indications of galectin-3 binding protein might have a role in innate immunity came when recombinant interferon alpha 2b was found to increase the expression and release of galectin-3 binding protein into a cell culture medium of breast cancer cell line CG5 and the concentration of galectin-3 binding protein in the serum of breast cancer patients. So, what is interferon alpha 2b? This is one of the cytokines, so it is an antiviral and an anti-neoplastic drug. So this is a drug that will stop cancer, right? And it is a recombinant form of the protein interferon alpha 2 that was originally sequenced and produced recombinantly in E. coli. So in the laboratory of this person. So basically, they were able to take interferon alpha 2 and then put it into E. coli, and then the E. coli secretes this into the environment, and then they can, market it and sell it, right, as a replacement for interferon alpha 2. So if we add this interferon alpha 2b, it increases the expression of galectin-3 binding protein in these breast cancer cell lines. And remember, these breast cancer cell lines actually produce a little bit too little of galectin-3BP. So interferon alpha, interferon beta, and interferon gamma mediated induction of either expression or secretion of galectin-3 binding protein has been later on documented by several independent research groups. These mediated induction of either expression or secretion of galectin-3 binding protein, and this has been later on documented by, this might be a typo of some form. I can't parse that easily. Just know that all these interferons have some role in the regulatory expression of galectin-3 binding protein. And these studies have also included identification of interferon regulatory factor binding elements from algal S3BP promoter areas. So this is saying is that these IFN regulatory factor binding elements are found on either the DNA or the mRNA of this galectin-3 binding protein, or at least in the promoter regions. So so the promoters are on the DNA, okay. And then I always mix up, so the DNA has the promoters, and then the RNA untranslated region will have, is it also called a promoter? One second, I need to. So they have the initiation codon, and then they have elements. I don't know what it's called, but I believe that there are, I think they're just called elements. So there are elements on the mRNA as well that can help initiate translation into the final protein. Regardless, I keep on forgetting biochemistry sometimes. Anyways, this, and these factor binding elements in the promoter area. So algal, the gene for galectin-3 binding protein, also appears responsive to tumor necrosis factor alpha, which can be inhibited with the nuclear factor kappa B inhibitor, which is this dihydroxymethyl epoxy quinomycin. This is in line with the presence of binding sites for nuclear factor kappa B transcription factors at the algal S3BP promoter area. So I believe, if I remember correctly, this tumor necrosis factor will cause nuclear factor kappa B activation, and this nuclear factor kappa B activation can theoretically bind to the promoter of this algal S3BP and result in the transcription of, Galectin-3 binding protein, or the mRNA for it. So it has also been reported that nerve growth factor, insulin, and thyroid-stimulating hormone-treated cells contain more, and hydrocortisone-treated cells less. Hydrocortisone being cortisol, they have less of this LGL S3BP mRNA. Interesting. But physiologic significance of these findings has remained elusive. So treatment with poly-IC, and remember this is the chemical that will imitate a double-stranded RNA and initiate a antiviral response in the cells that come into contact with it. This mimics the viral double-stranded RNA, and transfection with double-stranded nucleic acids, but not single-stranded nucleic acids, from various sources such as from mammalian cells, viruses, and bacteria. This will lead to higher LGL S3BP mRNA levels in the cell. So what we're saying here is that you will have higher mRNA levels of the galectin-3 binding protein if you trick that cell into thinking that there is an infection. So signals that either mimic or are characteristic for an ongoing infection and upregulate LGL S3BP transcription are summarized in Figure 2, and this is the figure. So, cellular innate immune responses mediated by galectin-3 binding protein. So, addition of galectin-3 binding protein into the culture of human PBMCs suppress. I don't know what PBMCs are. One second. The peripheral blood mononuclear cells. So that is in this particular part of the fraction. These cells consist of the lymphocytes and monocytes, whereas the erythrocytes and platelets have no nuclei, and the granulocytes. So yeah, it's basically just the lymphocytes, whereas everything else seems to go down into different areas. So the PBMC suppresses through as yet uncharacteristic or uncharacterized mechanisms. The transcriptional activity and secretion of these interleukins. So if you add Gal3BP to these lymphocytes, it ends up suppressing the transcriptional activity of these interleukins, which I believe these might be, well, these are cytokines. Granted, I think these are anti-inflammatory cytokines. Oh, wait, no, they are the hallmark cytokines of type 2 inflammation. So they are preventing the transcription of these interleukins, which are cytokines of type 2 inflammation. So accordingly, it was hypothesized that galectin-3 binding protein has a role in asthma, as the authors also evidenced a negative correlation between systemic concentrations of Gal3 binding protein and counts of eosinophils, which are the major effector cells of allergic inflammation. Moreover, galectin-3 binding protein interacts with dendritic cell specific intercellular adhesion molecules, also known as DC sign, in a calcium and carbohydrate dependent manner, and suppresses the DC maturation. So this is interesting. So galectin-3 binding protein can interact with these DC sign molecules, and it prevents them from maturing. Now, granted, this is galectin-3 binding protein that is going to be floating around in the solution, and it prevents these from maturing, and this prevention of maturation would theoretically lower the inflammatory response, because dendritic cell maturation ends up causing a more pro-inflammatory response. It also is the mature forms of dendritic cells will also mature the T cells into T helper cells. So this would be a way to prevent T helper cell formation. Interesting. So the study was conducted in a cancer context, and the authors proposed that the Gal3 binding protein DC sign interaction contributes to the development of tolerogenic microenvironments for galectin-3 binding protein overexpressing colorectal cancer cells. So this might theoretically be a way for the cancer cells to be able to suppress the immune system in that area. And so tolerogenic means the ability to create tolerance for, or generates tolerance, right? So if these cancer cells, they might be overexpressing galectin-3 binding protein in order to prevent the dendritic cells from maturing. And by preventing the dendritic cells from maturing, you end up creating an immunosuppressed site, and that might protect the cancer from the immune system. So binding of galectin-3 binding protein to DC sign has also been reported to suppress the differentiation of the monocyte-derived fibrocytes. So these are fibrocytes that come from the monocytes, which are a type of white blood cell. So let's look at what a fibrocyte. So these are mesenchymal cells that arise from monocyte precursors, and they are present in injured organs and have both inflammatory features of macrophages and the tissue remodeling properties of fibroblasts. So these cells are able to remodel tissue, so they are probably able to produce some form of they're able to remodel, and they can create activated connective tissue cells characterized by synthesis of proteins of the fibrous matrix. So they are able to create more of this fibrous matrix through activation. Interesting. So if you have more of these fibrocytes, which come from the monocytes or are differentiated from the monocytes, then you are more likely going to you're going to have more of this fibrous matrix being produced, right? So this suppresses that differentiation, so you will have less of this fibrosis or less of this tissue remodeling. Interesting. So further indicating that galectin-3 binding protein may have an immunosuppressive function. So I should say it seems as though it is improving. High levels of galectin-3 will be improving the innate immune response, right? Because it's pro-inflammatory, it's being activated upon these innate immune systems, right? But it seems to prevent the maturation of dendritic cells, which that would be a part of the adaptive immune response. So these dendritic cells, these are sort of like the teachers of the lymphocytes, right? They are going to, the lymphocytes being the white blood cells that are associated with the lymphatic system. So these, well, and that might be an extreme oversimplification, but these dendritic cells, they can exist in their immature forms where they are telling all the cells around it that you really shouldn't worry about the environment. The environment is safe, so you don't need to be activated. However, when these dendritic cells have matured, they are then telling all the cells that whatever antigens are present in this environment are dangerous, and therefore it activates the immune system to search and destroy for those particular antigens. So it sounds like galectin-3 BP is telling the environment, do not use the adaptive immune response, use the innate immune response, which to me makes sense. However, that would be very bad in, say, a cancerous situation, right? Because cancer, that is not a viral infection, right? And that is not a bacterial infection. So you do not want you wouldn't necessarily want the innate immune response. Well, granted, I take that back, because these cancer cells are also going to be expressing a lot of damaged proteins on their surfaces, so the innate immune response might also be able to identify the cancer cells in that particular way. But you would really also want the adaptive immune response to come in and target that as well. I would have to do some reading on it, but to me what is sounding, the model that's sort of forming in my head is that you have this galectin-3 binding protein that is causing more expression, or causing the immune system to be more innate based rather than adaptive based. So 1, 14. So 1 second. The first indication that galectin-3 binding protein may have immunostimulatory functions came when galectin-3 binding protein was used to precondition human PBMC cells. So these are those leukocyte cells, and their cancer killing activity was significantly increased. So yeah, I take back what I said earlier. It sounds like the galectin-3 binding protein might be able to increase the cancer cell killing activity of those lymphocytes. And this was measured by the cell death of the natural killer cell sensitive K562 cells, and in parallel with lymphocyte activated killer cell sensitive Dalby cells. So these are just different cancer cells, right? Cancer cell lines. And we know that they are sensitive. These we know are sensitive to the natural killer cells, and these we know are sensitive to the lymphokine activated killer cells. So cell enrichment and depletion studies of PBMC showed that CD14 positive cells, which are the monocytes and macrophages, they produce interleukin-1, interleukin-6, granulocyte macrophage colony stimulating factor, also known as GM-CSF, and tumor necrosis factor alpha, as well as displayed higher amounts of ICAM1 on the cell surface upon Gal3 binding protein treatment. So after you've added Gal3 binding protein, and remember this is of the monocytes and macrophages, they started producing these pro-inflammatory cytokines, interleukin-1 and interleukin-6, and then this granulocyte macrophage colony stimulating factor. This is just another cytokine that is, let me see if it has a good, okay, so unlike granulocyte colony stimulating factor will specifically promote neutrophil proliferation and maturation, and these are the neutrophils. So these are a type of granulocyte that they're the most common white blood cell in the body, if I'm remembering correctly. However, this granulocyte macrophage colony stimulating factor will affects more cell types, and it is especially true for the macrophages and the eosinophils. So this GM-CSF is more broad than the neutrophils. So when we add this galectin-3 binding protein, it ends up activating, and we add it to these lymphocytes. They ended up causing the expression of interleukin-1, interleukin-6, and this granulocyte macrophage colony stimulating factor in the CD14 positive cells. And the same goes for tumor necrosis factor alpha. And as well, they displayed higher amounts of ICAM1 on the cell surface. So what is this ICAM1? So we need to break down everything. So it is also known as CD54. And it encodes a cell surface glycoprotein, which is typically expressed on endothelial cells and cells of the immune system. And it binds to integrins of these type. And it's also exploited by rhinovirus as a receptor for entry into respiratory epithelium. So this might be important. So this is something that is also on endothelial cells. It's not just on the white blood cells. So they are a member of the immunoglobulin superfamily. And the superfamily of proteins including antibodies to the T cell receptors. So I think I've read this one before. So here's the function. So the protein encoded by this gene is a type of intercellular adhesion molecule continuously present in low concentrations in the members of the leukocytes and the endothelial cells. Upon cytokine stimulation, the concentrations greatly increase, and ICAM1 can be induced by interleukin-1 and tumor necrosis factor, and is expressed by the vascular endothelium, macrophages, and lymphocytes. ICAM is a ligand for LFA1, which is the lymphocyte function associated antigen 1, which is a receptor on leukocytes. And when activated, the leukocytes bind to endothelial cells and then transmigrate into tissues. So LFA1 has been found in a soluble form, which seems to bind and block ICAM1. So if you take this LFA1 and it's in solution, you can actually block its ability to bind with the surface LFA1. So it is an endothelial and leukocyte-associated transmembrane protein, long known for its importance in stabilizing cell-cell interactions. Characterized as a site for the cellular entry of human rhinoviruses. So this is something that can allow rhinoviruses to infect your endothelial cells of your nose. And because of these associations with immune responses, it has been hypothesized that ICAM1 could function in signal transduction. And it produces pro-inflammatory effects such as inflammatory leukocyte recruitment by signaling through cascades involving a number of kinases. So it also, ICAM and soluble ICAM have an antagonistic effect on the tight junctions forming the blood-testes barrier, so they thus playing a major role in spermatogenesis. Okay, so I think I'm not going to go too far on this. Just know that this is on the surface of some cells, and it promotes adhesion, and it promotes the leukocytes to move into or dig into the endothelium. So, and sorry, I need to, so leukocytes, those are any of the white blood cells. So maybe they are convincing the neutrophils to dig in. I'll have to do more reading on that. So there was another paper that I went over earlier that talked about how Galectin-3 binding protein and Galectin-3, the co-localization of those actually causes some of the, the neutrophils to dig into the epithelium, or dig into those endothelial cells. So that might be a similar process. I don't know, because immunology is kind of really freaking complex and I hate it sometimes. So these changes were experimentally linked to activation of the CD3 positive T cells in these PBMCs, including the CD3 positive T cell mediated secretion of interleukin-2. And this is a potent autocrine inducer of effector functions in T cells. So autocrine is like a hormone where the cell will release it, or it basically is a hormone of the T cell that activates its own self. So, for example, and it's a potent autocrine inducer of effector functions of T cells, for example, against cancer cells. In part, T cell activation is supported by a positive correlation detected between Gal-3 binding protein and soluble interleukin-2 receptor. And this is a marker for T cell activation. So concentrations in serum samples of ovarian cancer patients. So Gal-3 binding protein induced interleukin-6 expression and secretion, which has been documented by several research groups in multiple different cell types. So, and it requires carbohydrate-mediated interaction between Gal-3 binding protein and Galectin-3 at the cell surface, and involves the MAPK and ERK pathways. So these are the MAPK and the ERK, those are just pathways that are within the cell. But as I was saying earlier, it requires, if Galectin-3 binding protein, this Galectin-3 binding protein is interacting with Galectin-3 in order to cause that effect to happen. And this is carbohydrate mediated. So this Galectin-3, which is a lectin, must be binding to the Galectin-3 binding protein somewhere. So, and it's binding through one of the carbohydrates. So remember, Galectin-3 binding protein is heavily glycosylated and glycosylated explicitly. And so that must be, or might be, where one of these galectin molecules are binding to. So exogenous Galectin-3 binding protein has also been shown to activate the THP1 cells alone and additively with interferon gamma, as judged by the increased concentrations of neopterin in the cell culture medium. So neopterin, this is a marker for the cell-mediated immune activation, and is secreted mainly by the monocytes and macrophages. So it has also been shown that the addition of Gal3 binding protein into the human PBMCs induces cytosolic influx of calcium. So this is a strong second messenger. Taken together, the current data indicates that galectin-3 binding protein has immunosuppressive as well as immunostimulatory functions in vitro. For most of the cellular responses, however, the molecular basis, for example, specific cell surface receptor or specific downstream signaling events, has remained elusive. So this is something that, kind of annoys me a little bit, is that we can find a protein that has all of these seemingly paradoxical or contradictory effects on the system, and it's so, like, as we were saying earlier, it seems that this galectin-3 binding protein is being upregulated for cancer to help protect itself from the immune system. But then at other times, it seems like it's being used to activate the immune system against cancer cells. So it seems like all this biology, I hate it sometimes, really. So experimental in vivo data of the systemic immunomodulatory functions of galectin-3 binding protein are available for cancer cell xenograft studies conducted in a thymic nude mouse. And I don't know what this is, and I'm kind of afraid to search it up on Google, but a thymic probably means without its thymus gland, right? Which is a genetic mutation that causes a deteriorated or absent thymus. So yeah, it's athymic, it doesn't have a thymus. And that inhibits the immune system because the thymus is where a lot of the thymocytes migrate from the bone marrow into the thymus so that they can mature into T cells, right? So the T cell is a type of lymphocyte. So that is basically the thymus is where the T cells are produced. And if you do not have any of these T cells, which is the case for this thymic nude mouse, there is a systemic immunomodulatory function of the galectin-3 binding protein, and they are available for cancer cell xenograft studies, which are conducted in thymic nude mice. First, the authors showed that when galectin-3 binding protein negative mouse mammary carcinoma cell lines, sorry, that's a lot of words, were transfected with the stably produced human galectin-3 binding protein, the cell lines became severely impaired in their ability to form solid tumors. So there are some tumors out there that are within, so you can bring these tumor cells and you can inject them into these mice, these thymic nude mice, right? And when you inject these cancer cells, and in this case the mouse mammary carcinoma cell line, you can run experiments on them by injecting these cancer cells into them. This particular cancer cell line does not have galectin-3 binding protein, but we know that it normally forms tumors. However, when we inject the human galectin-3 binding protein, it impairs the ability for them to form these solid tumors inside of the mice. So this tells us that this human galectin-3 binding protein is being suppressed in this particular tumor. So when galectin-3 binding protein MM5 transfectants were xenographed into the left leg, the authors observed a significant suppression of the MM5 xenografted into the right leg as compared with the control animal group xenographed with MM5 alone. So when they take galectin-3 binding protein and they transfect it into the MM5, so now that this MM5 normally doesn't produce any galectin-3 binding protein, but now it does when they transfect it, they xenografted it into the left leg of these mice, and the authors observed a significant suppression of the MM5 xenografted into the right leg as compared with the control animal groups xenographed with MM5 alone. So this is interesting. If we take a rat and if we give the rat a transfection of this galectin-3 binding protein MM5 in one leg and then transfect it and then also infect it with the normal MM5, in the other leg, the galectin-3 binding protein in one of the cancers ends up affecting the other cancer and makes the other cancer suppressed. So that's interesting. So even the galectin-3 binding protein in one cancer is able to affect the cancer in the other leg. However, if you have both legs infected with just the MM5, they are both progressing like normal cancers do. So this indicates that galectin-3 binding protein induces systemic anti-tumor effects, possibly through immune system activation. So indeed, galectin-3 binding protein overexpressing xenografts had more natural killer cells and macrophages as compared with controls. Moreover, the tumor vasculature of galectin-3 binding protein overexpressing xenografts, and I should say the xenografts are when you graft tissue into a living organism. This is strongly ICAM and VCAM1 positive. So so the tumor vasculature of galectin-3 binding protein had a lot of these ICAM1s and VCAM1s, which, as we said earlier, would encourage the white blood cells to dig into them, right? And so a tumor that sends a signal to the white blood cells saying, dig into me, that's probably going to be a tumor that is going to be more difficult. It's going to be more difficult for them to survive because the white blood cells are activated and digging into them. So it is also remarkable that the addition of galectin-3 binding protein into cell cultures induces, through a yet uncharacterized mechanism, major histocompatibility complex 1 transcription and increases the amount of MHC1 protein at the cell surface, almost to a level comparable seen after interferon gamma stimulus. So this galectin-3 binding protein will upregulate this major histocompatibility complex type 1, which is one of the proteins that basically major histocompatibility complex will have different peptides that are synthesized or come from a degraded protein. Those are going to be added to the major histocompatibility complex, and then the major histocompatibility complex will then be moved to the cellular membrane, and then the cellular membrane is going to then have these peptides that are being presented to the environment on top of these MHCs. So the fact that galectin-3 binding protein causes that would be an indicator that it is affecting the what would the word be? The expression of, or the activation of the immune system. So imagine you are a cancer cell, right? And a lot of your proteins are damaged, mutated, and do not look the way that they should. And so if the major histocompatibility complex presents a mutated protein, then that mutated protein will then have a peptide that is mutated that is presented on the surface of it, and that can warn the immune system, oh shoot, there is some unknown protein that is coming out of the cell. Therefore, this cell either is infected with the virus, or it is a cancer, and therefore I need to attack this. So yeah, that would be, so as the loss of MHC1 expression is common in malignant cells, the authors propose that galectin-3 binding protein could tamper with the tolerogenic tumor microenvironment. The proposed immunomodulatory functions of galectin-3 binding protein identified in non-infectious experimental settings are summarized in figure 2. So so a lot of cancers do not express major histocompatibility complex 1 at all. And this is one of the ways that they are able to avoid the immune system, right? So galectin-3 binding protein would normally, allow, or what would the word be? It would normally increase the major histocompatibility class 1 on the surfaces of these cells, which would theoretically make them more vulnerable to immune cells that recognize these strange epitopes or these weird peptides. However, since some of the cancer cells do not have any MHC, it doesn't matter if there's galectin-3 binding protein present. And so if there's galectin-3 binding protein that is being upregulated, that can screw up with the immune system while still leaving the cancer A-okay, right? Regardless, we should keep on going. So galectin-3 binding protein in viral infection. So first indications of the role of galectin-3 binding protein in viral infections were obtained when a group of scientists observed patients with HIV-associated Kaposi sarcoma to have unusually high galectin-3 binding protein serum concentrations. Okay. The authors further observed that there was a negative correlation between the galectin-3 binding protein concentrations and the CD4 positive cells. So these are the T helper cells, which are the targets of HIV. So the CD4 is the receptor that HIV uses to get into these cells. Anyways, but a positive, let me get a drink first. Anyways, a subsequent retrospective study with HIV-infected patients showed that increases in galectin-3 binding concentrations preceded the decrease in the CD4 positive cells, which is suggesting a prognostic value for galectin-3 binding protein concentrations in HIV infection towards AIDS. So as people are moving or progressing towards acquired immunodeficiency syndrome, then this galectin-3 binding protein increase might be an indicator that this is happening. So indeed, high galectin-3 binding protein concentrations in HIV-infected individuals were associated with faster progression towards AIDS in follow-up studies. So the higher this was, the faster they progressed into AIDS. So the galectin-3 binding protein concentrations positively correlated with HIV RNA concentrations in HIV-positive children and adults undergoing HIV therapy. Increasingly, or interestingly, higher Gal3 binding protein concentrations have been observed in HIV-positive mothers giving birth to healthy infants as compared with HIV mothers with HIV-positive infants. So what they're saying here is that the mothers who are producing more of this galectin-3 binding protein, their children were less likely to become infected with HIV. So what this is indicating to me is that the galectin-3 binding protein isn't causing the negative effects of AIDS. Instead, it is being produced or upregulated to help protect the person from AIDS. However, the more protectant that you are excreting into the environment is probably an indicator that your disease progression is happening pretty rapidly. So this is indicating that galectin-3 binding protein may have antiviral activity. So mechanistic insights on the potential antiviral role of galectin-3 binding protein in HIV infection came when Loimaranta and colleagues observed that galectin-3 binding protein overexpression reduced the amount of mature GP120 and GP41 in released virions. So these are just different proteins that make up HIV. So GP120, this is the envelope glycoprotein of HIV, and GP41, this is the subunit, it's another subunit of the envelope protein complex of retroviruses. So just know that that is what's happening. And in fact, let's go to this Wikipedia article, and it might have, yeah. So here you can see, let me zoom in. So this is the CD4 receptor on your CD4 positive T cells, and this GP41 is one of the envelope proteins that basically anchors this whole thing to the cellular membrane, or the envelope membrane of the HIV. And then this GP20 is attached to the GP41, and this interacts with the CD14, and this whole thing helps the virus get into the cell. So as you can see here, this GP120 will then interact with this CCR5, and then this entire process ends up resulting in the HIV DNA, or sorry, the HIV RNA allows the whole thing to enter the cell. So if you overexpress Gal3 binding protein, you end up reducing these virions, or reducing the mature GP120 and the GP41 in the released virions. So increasingly, interestingly, the amount of the released virions was not altered drastically, but rather the processing of GP120 on the GP120 envelope into GP160 envelope into GP120 and GP41 proteins was impaired. So what's happening is GP160 is being synthesized by the ribosomes, but this GP160 needs to be proteolytically cleaved into the final GP120 and the GP41 proteins. So if you remember, one of my first videos was discussing proteolytic cleavage and why proteolytic cleavage is important to viruses. So if you want, you can go to the beginning of the playlist to see which one. I think it might have been the second or third video that I did. However, this leads to reduced, if you have high levels of this Gal3 binding protein, you actually have reduced levels of cleavage into the final GP120 and GP41 proteins. And this also leads to reduced infectivity. So that means this is indicating that Gal3 binding protein is protecting us from viruses. So even though it's very highly expressed in the HIV patients that end up getting more serious diagnoses, that it's actually because it's trying to protect you, not because it's causing it. Anyways, importantly, the SI, small interfering RNA-mediated knockdowns of endogenous Gal3 BP enhances the incorporation of GP120 and GP41 proteins into progeny virions and increases their infectivity. So if you downregulate Gal3 BP, you end up creating more viruses, or more infectious viruses. So the authors demonstrated with Gal3 binding protein transfection experiments and by the neglectable response after Gal3 binding protein exogenous addition that the impairment of HIV infectivity occurs inside the cell. By truncating Gal3 BP, the authors demonstrated that the central BTB-POZ and back domains, which is this particular fragment, this mediates the observed interference in the GP160 envelope maturation. So it doesn't require the entire Galectin-3 binding protein, it just requires this BTB-POZ and back domains, which is between 127 and 409. Interesting. Yeah, okay, sorry. I think earlier I might have said N-terminal is at the beginning. I meant to say it's at the end. But sorry, I'll look back on the video and make note of that. So the authors propose that the likely mechanistic explanation relates to the interference of the, what's this? The furin-mediated GP160 envelope processing. So furin is one of the proteolytic enzymes that is able to break apart these HIV proteins into other proteins. So furin is one of the proteases. And it's not the only protease that can do this, I believe, but it is one of the best at doing this. So this BTB-POZ domain might be able to interact with furin, and it can interfere with the furin-mediated processing either sterically, which is when it literally doesn't allow other things to fit in, or through trapping the GP160 envelope into the endoplasmic reticulum, thus hampering the GP160 ENV entry into the furin-positive cellular compartment. However, the authors were unable to detect interactions between GP160 envelope and Gal3 binding protein, so they weren't able to see a direct interaction that way, and therefore the mechanistic explanation remains elusive. Interesting that they mentioned that. I might have to look into that as well, that if galectin-3 binding protein is able to somehow interrupt this processing of proteins, that would be very, very interesting. I'll really have to look into that. So a more recent study from another group confirmed the findings of Loimaranta and colleagues on the ability of galectin-3 binding protein to interfere with this processing. But they also showed pronounced inhibition of virion release. They also found that not only the central BTP pause and back domains, but also the SRCR domain was able to reduce HIV particle infectivity. So it seems like the whole, even though it's not necessary to have SRCR to reduce HIV activity, it seems as though both of these domains are able to reduce HIV infectivity, which is very good to know. So the authors further observed that Gal3BP reduces the incorporation of HIV GAG into the progeny virions. So interestingly, Gal3 binding protein was shown to interact with HIV GAG and cytoskeletal compartment vimentin. Component vimentin. So the authors proposed that formation of this 3-protein complex reduces the amount of GAG and the plasma membrane, and therefore would also inhibit the formation of progeny virions. Taken together, Gal3 binding protein appears to elicit an antiviral effect inside the HIV-infected cells, affecting both the release of progeny virions as well as their infectivity. Okay. So the observation that the proportion of HIV-negative intravenous drug users had unusually high serum concentration of Gal3BP prompted Artini and co-investigators to study whether or not Gal3BP upregulation would be associated with other chronic viral infections. And indeed, the authors found high Gal3BP concentrations in the serum of hepatitis B and C virus infected individuals. The concentrations of Gal3BP was found to be significantly higher among this HCV, hepatitis C, as compared with hepatitis B infected patients. And furthermore, the galectin-3 binding protein concentrations of hepatitis C patients were found to correlate with severity of liver damage. So the higher the galectin-P concentrations you had, the more liver damage you had. And remember, hepatitis is named hep, that would mean of the liver. So hepatitises target the liver and damage the liver more so than other organs. And so the people with more liver damage had more of this galectin-3 binding protein in their serum. So a galectin-3 binding protein glycovariant that binds to Wisteria floribunda lectin. So frequently we use lectins, which are carbohydrate binding proteins. We frequently will have a variety of lectins from a bunch of different sources. The Wisteria floribunda, which is Wisteria, they have a lectin that is frequently used in these glycoprotein studies. This is suggested to function as a serum marker for the severity of hepatitis virus-induced liver fibrosis, or the scarring of the liver, for example, in reference 77. Additionally, patients with the high galectin-3 binding protein concentrations were found to respond poorly to interferon alpha therapy. This was confirmed by a follow-up study showing that patients with non-1b HCV having low HCV RNA and galectin-3 binding protein concentrations, they are most likely to respond to interferon alpha therapy. So if I remember correctly, the interferon alpha is going to help release or cause the production of more galectin-3 binding protein, or the other way around. I can't remember exactly what's going on, but if you already have high levels of the galectin-3 binding protein, it probably means that you are already exhausting your interferon alpha pathway, right? It's already saturated, so it's not going to help anymore. So also, the observation that galectin-3 binding protein might be a predictor of the hepatitis C infection severity was later confirmed by an independent study. So interestingly, the galectin-3 binding protein concentrations induced by different HCV genotypes vary, and highest concentrations are seen in patients with genotypes 1 and 4. So in the same study, a sustained response to HCV antiviral therapy was more often seen in patients with lower Gal3 binding protein concentrations. Oh, so they're saying if you have low Gal3 binding protein, you responded better to therapy. The authors speculated that INF alpha 2b therapy, which is known to induce Gal3BP, so yeah, that was right. The interferon alpha normally upregulates Galectin-3 binding protein. But if you already have that Galectin-3 binding protein expressed as high as it can, then interferon alpha probably isn't going to be helping you that much more. Anyways, this would be more efficiently. Oh, that would be a more efficient trigger for Galectin-3 binding protein mediated T cell activation and/or elevation of interleukin concentrations in patients with lower initial Gal3 binding protein concentrations. So they're saying here is if you have low Gal3 binding protein, this interferon alpha might be a good therapy to help increase that. Taken together, viral infections also, other than that, caused by HIV may increase systemic concentrations of Galectin-3 binding protein. So the acute viral infection. So the first indication that of Galectin-3 binding protein upregulation in acute viral infections came when Vuokko and colleagues were studying systems level changes in gene expression of muscle cells as a result of dengue infection. Gal3BP upregulation occurs also at a protein level in dengue patients' serum, along with the galectin-9 upregulation. Same thing, also hantavirus infection and Epstein-Barr virus. And let's see, let me go down. I don't know if I should, you know what, I'll read everything. I might as well. I made it this far. So the authors found that human, but not mouse Galectin-3 binding protein efficiently bound the recombinant adeno-associated virus 6 and was also able to interact with recombinant adeno-associated virus 1 and 5. So the addition of Gal3BP to RAAV6 preparations was shown to induce aggregation of viral particles and to impair the RAAV6 transduction ability into a mouse model. So around the same time, Vuokko and colleagues observed that Gal3 binding protein co-purifies with hantavirus particles, likely through direct interactions between hantavirus glycoprotein and Galectin-3 binding protein. So it sounds as though this Galectin-3 binding protein is able to interact directly with these virus particles. So the finding led authors to study Galectin-3 binding protein concentrations in individuals with mild forms of hemorrhagic fever with retinal syndrome, and indeed they found Galectin-3 binding protein concentrations to be elevated during acute phase of illness. So the authors further employed archival samples of experimental hantavirus infection done on this macaque and found that Galectin-3 binding protein concentrations to correlate with serum creatinine, tumor necrosis factor alpha, and interleukin-10 concentrations. So the authors demonstrated that hantavirus infections on HUVEC induces Gal3 both at the mRNA and protein level, and that small interfering RNA-mediated knockdowns of Galectin-3 binding protein increases the virus replication in the infected cells. And in human patients, Gal3 binding protein was found expressed in tissues which are targets for virus replication during infection. So Gal3 binding protein concentration correlates with parameters reflecting the severity of the disease, but also with the maximum concentration of serum terminal complement complex SC5b9. So taken together, these findings suggest that Gal3 binding protein might be locally produced and released in acute viral infections, leading to activation of antiviral innate immune responses. That would make sense to me. Almost done, so great. So the Gal3BP in bacterial infections. So as compared with viral infections, publication of Gal3BP in bacterial infections are scarce. So one recent report describes elevated serum Gal3BP concentrations in patients suffering from blood culture positive bacterial infections. So these people have reported that Bartonella effector protein, also known as BAP-A, which is delivered into the host cytosol through VIRB type 4 secretion secretion system associates with Gal3BP. So I would assume Bartonella is a bacteria, and it has an effector protein also known as BEP-A, and it delivers. This protein is delivered into the host cell cytosol through this secretion system. So functional relevance of this finding is not yet known, but it implies that Galectin-3 binding protein might have intracellular functions in innate immune responses against invasive bacteria. So extracellular functions for Galectin-3 binding protein in bacterial infections have been reported. Mice that are deficient of Galectin-3 binding protein, also known as CyCap, in mice, they show increased susceptibility for lethal effects of intraperitoneally administered lipopolysaccharides. So lipopolysaccharides, these are polysaccharides associated with some form of lipid that different bacteria or different pathogens will produce, and they will frequently activate the inflammatory response. Sometimes if you administer the LPS directly, this can cause very bad inflammatory damage because it activates the immune system so much. So mice that are deficient of Gal3BP are even more susceptible to this intraperitoneally administered LPS, and that would be an injection basically into the internal organ area. So the highly abundant molecule released by and found on the outer membrane of gram-negative bacteria. So that's, yeah, the gram-negative bacteria have a lot of this LPS. So Gal3BP deficient mice are apparently healthy, fertile, and their blood counts are in normal range, except that they spontaneously develop colonic mucosal hyperplasia early in life. So what is colonic mucosal hyperplasia? So this is, let me get a Wikipedia article up that might help you. So this is hyperplasia. First, you have normal cells, and then the normal cells become hypertrophic in that they grow a lot, and then, or you could have hyperplasia where you have a bunch of smaller cells. So that would be the difference between hyperplasia and hypertrophy. So this would be hypertrophy where the cells become bigger, and that can cause damage if the cells become too big. Or you could have hyperplasia, which could cause damage because you have too many cells and they're stretching this basement membrane. So yeah, and may lead to gross enlargement of an organ. So this would be hyperplasia in the colon, and I presume the mucosal lining, which would not be good. So the LPS stressed Galectin-3 BP deficient mice display elevated concentrations of pro-inflammatory cytokines, either systemically, interleukin-12 and interferon gamma, or locally through tumor necrosis factor alpha. So in the peritoneal cavity as compared with the wild-type mice. So peritoneal exudate cells, which are mostly macrophages from both the wild-type and the Galectin-3 BP deficient mice, produced more tumor necrosis factor alpha upon LPS stimulation in vitro when the cells were incubated in the presence of Galectin-3 BP deficient serum as compared with the Galectin-3 BP containing serum. Yeah, so basically Galectin-3 BP seems to have this anti-inflammatory response as well. So this data, or the data indicate that Galectin-3 binding protein acts specifically to down-modulate the LPS signaling through an extracellular regulatory mechanism. The observation that Galectin-3 BP is retained on beat-immobilized CD14 during chromatography of, I lost the recording since I went over 2 hours. 1 second. And however, the mechanistic understanding has remained elusive. For example, Gal3BP does not appear to directly bind the lipopolysaccharides, and experimental evidence for direct interactions with CD14 and Gal3BP is lacking. Immunosuppressive activity of Gal3BP in lipopolysaccharide signaling might involve indirect effects through other serum and or cell surface components. So, I mean, this makes sense. So Galectin-3 binding protein seems to be able to interact with a lot of the cell surface components. So its effects are going to be completely dependent on what is on the surface of the cells nearby. For example, Gal3BP could signal through cyclophilin C, which is the target of the immunosuppressive drug cyclosporin A. So that is interesting, and I think I need to look into that more. Granted, I think it was cyclophilin A that I had done work on. So cyclophilin A, one of the cyclophilins interacts with the CD147. So let me, CD147. Yeah, so it's cyclophilin A, not cyclophilin C that interacts with CD147. So sorry about that. So dead end. But maybe, I mean, they are conserved, so there might be some crossover there. Anyways, the proposed binding partner of the mouse Gal3BP. So for example, Gal3BP could signal through cyclophilin C, which is a target for the immunosuppressive drug cyclosporin A, and proposed binding partner of the mouse Gal3BP. Recent report demonstrates human Gal3BP to have an immunosuppressive function against lipopolysaccharide-stimulated neutrophils by inhibiting both spontaneous and lipopolysaccharide-induced production of reactive oxidant species by the neutrophils. So the inhibitory activity depends on the sialic-dependent interaction between, and remember, sialic acid, this is one of the glycosylation events that has taken place on one of the N-terminal parts of the N-terminal is the beginning. Oh man, I keep on mixing up N and C terminals, but remember, the sialic acid is part of this in some way. So, and this will bind to the inhibitory activity depending on sialic acid-dependent interactions between Gal3BP and the sialic acid binding lectin-9. So sialic acid binding lectin-9 will bind to the sialic acid that is on this Gal3BP. So, and this is also present on the surface of neutrophils. So these neutrophils will have this Siglec-9. So taken together, the current data imply that Gal3BP is part of a negative feedback loop to suppress the inflammatory response during infections with gram-negative bacteria. So here are the concluding remarks. So Gal3BP is a ubiquitous and multifunctional secreted glycoprotein of the SRCR domain-containing protein superfamily. And you know what? I've already said enough. So with that, I just want to say that it sounds like this Gal3BP plays an important role at determining what type of immune pathway we are going to be working with. And so we will theoretically, the upregulation of Gal3BP might be very beneficial against viral infections. However, it seems to be hit or miss on cancerous growths, right? It appears as though the cancerous growths that are immune usually are missing the major histocompatibility complex 1, which prevents them from expressing more of it in response to Gal3 binding protein. So with that, I think I'm going to go up to this, let's see. Yeah, let's go up to this, what would the word be? Figure. And let's look at the different things that Gal3 binding protein can do. And remember, Gal3 binding protein can actually form rings and dodecamers. So that might be something that is of importance as well. So here we have, in the nucleus, we have the different factors or the different cytokines or different chemicals that can cause this algal S3BP, the gene, to create the mRNA to create the Galectin-3 binding protein. So all these things have the ability to upregulate it, and they seem to upregulate it through this IRF element. And the IRF element is I forget the bind IRF. Okay, it's just one of the elements. Let me look up element. Oh, come on. I really wish the interferon regulatory factor binding element is what it is. So the interferon regulatory factor binding element. So that's what this IRFE is. And that's what these interferons are going to be interacting with in some way. I don't know if it's directly or through some sort of cellular signaling. Regardless. So for instance, in viral infections, we can have the Galectin-3 binding proteins interact with the virus itself, possibly, right? So that's one thing that the Galectin-3 binding proteins can do. And keep in mind, this might be able to cause clustering, because remember, the Galectin-3 binding proteins can also interact with Galectin-3s, and Galectin-3s help participate in clumping things together as well. So this might be a way to clump a bunch of viruses together. So, and here on the inside, this is talking about the production of the GP120 and the GP41 for producing HIV, right? And so here it seems like Galectin-3 binding protein is able to prevent this from happening somehow. So that's for the viruses. For the let's go to this normal cellular signaling. So one, you can have all of these different factors that can either downregulate, so like the interleukins 4, 5, and 13 can downregulate, the Galectin-3 binding protein can downregulate the interleukins 4, 5, and 6, but then upregulate tumor necrosis factor alpha, GM, CSF, and interleukin 1. So that's one way, and granted, this is not known how it does it. So two, Galectin-3 binding protein in interaction with Galectin-3 can cause this cascading pathway, which then causes transcriptional changes within the nucleus, and this transcriptional change ends up producing more interleukin-6, which is also a pro-inflammatory cytokine. Galectin-3 is, through some unknown mechanism, able to increase intracellular calcium, which could be a signal for a range of different calcium effects. The Galectin-3 binding protein can also cause the increase of production through some unknown mechanism, the production of these ICAMs, which these ICAMs are important in signaling to the immune cells nearby that they need to, say, move to that location, or they can cause clustering. They can do a bunch of different things. Gal3BP can also upregulate these major histocompatibility complexes 1, which can help the immune cells discover or strange peptides that are coming from within the cell, and that could be virus related, or that could be pathogen related, or it could be cancer related. So cancers will also have mutated proteins as well. So that can happen. We also have this, the DCSIGN, which I forgot exactly what that was, but I'm getting a little tired of reading. Just know that the Galectin-3 binding protein can interact with it and then cause some sort of downstream signaling. And then for bacterial infections, what Galectin-3 binding protein can also do is it can inhibit CD14, which inhibits its ability to cause this crazy response in response to these lipopolysaccharides. So that is one way to inhibit it. And then Galectin-3 binding protein will also interact with Sigleg-9, which will then prevent these reactive oxygen species from being produced from lipopolysaccharides. So remember, these lipopolysaccharides would be an indicator that you have gram-negative bacteria in the environment. And so if you straight up inject it, that actually puts the immune system into overdrive. However, this Galectin-3 binding protein helps modulate or prevent that from going into overdrive. So with that, I'm going to head on out, and hopefully this gives you a broad understanding of what Galectin-3 binding protein can do. And then, since we know that Galectin-3 binding protein is upregulated in COVID-19 patients, as with other viral infections, this might be able to help us figure out what the hell is going on. So with that, thanks for listening, and hopefully I will see you later today.